So what just happened? Sometimes, cells need to degrade or destroy very specific proteins. In order to do this, they have to first find that protein amidst all the ones it has, and then destroy it. How does it do both? It uses the ubiquitin-proteasome pathway. So first, what is ubiquitin? Well, it's a protein that's found ubiquitously in eukaryotic cells, or cells with nuclei. It's also special because it serves as a marker for degradation. Think of it as a flag of death for proteins. And this process of marking proteins for degradation is called ubiquitination. But how does it work? Let's imagine each protein has a side of a puzzle piece. Each puzzle piece has a unique shape, and so does every protein. Here's ubiquitin. We'll imagine it's a sticker. So the first step in ubiquitination involves an enzyme named E1. This guy activates the ubiquitin. Think of it like peeling the sticker. Then comes E2, who conjugates with the ubiquitin. Think of it like the sticker getting put on E2. And now comes E3. Now here's a special thing about E3. There are actually hundreds of different kinds, each with a unique shape. Remember how each protein also has a unique shape? So if the cell wants to degrade a very specific protein, it can just send out the E3 it knows matches with that protein. So, once the E3 has found the target protein, it pulls in the E2, which has the activated ubiquitin, and then sticks the ubiquitin onto the target protein at a specific linkage site, usually the K48 lysine residue. Sometimes this only happens once, and there's only one sticker on the target protein. This is called monoubiquitination. Other times this process is repeated, forming a chain of ubiquitin, and this is called polyubiquitination. And once this has happened, the proteasome comes in, a complex barrel-shaped structure that degrades these polyubiquitinated proteins. You can think of them as shredders. So, why do cells do this? Well, for one thing, they aren't perfect, and sometimes they produce faulty proteins which need to be destroyed. But another thing is that proteins don't last forever, and since the cell constantly produces new proteins, it has to keep degrading the old ones to maintain a balance of just enough proteins, or homeostasis. It's not an evil process. It's a necessary and amazing part of proper cell regulation. And it's even happening right now in you and me.